All right. Hello, everybody. Welcome to Nerd Nite. Woohoo! Nerd Nite! It's an appropriately rainy April Nerd Nite. Thank you for coming out and braving the rain. My name is Lauren. I'm with the Ann Arbor District Library, and we're super excited to have you here for a night of awesome nerdy talks. So before we get into it tonight with our speakers, there we go. I wanted to give you a quick origin story of Nerd Nite. This is Chris. He's a graduate student, and long ago, Chris was in grad school, and in his when he wasn't studying and doing his research, he was at the Midway Cafe in Boston doing his drinking, which is also important. It probably helped a lot. So he researched. His research took him across the world to Cameroon, where he was studying this beautiful indigo bird. Aw, so cute, right? Yeah, absolutely adorable, innocent in all ways, indigo bird, but actually not really. It's a jerk bird. The indigo bird is a stone cold killer. It's one of those cowbirds which infiltrates a nest of another bird, right? And then lays its eggs, and then the indigo bird's eggs hatch and proceed to eat the other babies. And the unsuspecting mother bird of the other species just raises this indigo bird as her own, and the vicious cycle repeats itself. So pretty cool. So Chris would regale anyone who would listen to him night after night at the Midway Cafe of all the nitty-gritty details of this indigo bird, until finally the bartender said, "Chris, stop. You got to stop. Okay, you get one night. Okay, we're going to clear the stage, and you get a mic, and you get to talk about your indigo birds one last time." Okay, and he did, and it was a big hit. In fact, everyone was like, "We should do that with other people telling their stories." And Nerd Nite was born, and it took over the world. And so we have Nerd Nites all across this map, and but ours is the best, of course, because it's sponsored by the Ann Arbor District Library. Woo! Yeah! And special thanks to our venue Live. Don't forget to tip your bartenders. Woo! Thank you so much for being here. We're going to kick things off tonight with Natalie Catchings. She is going to, she's been doing science her whole career, and that has involved taking DNA from weird stuff, right? Yeah, like mammoths, I think I read. Yeah, okay. And COVID and crime scenes. So pulling useful info out of those things. I'm excited to hear about this. But she'll also be talking specifically about how science is portrayed, right, in movies and TV, and whether or not we should trust those representations of science. I think I heard that's maybe not. Okay, all right. So we're going to have Natalie on the stage now. Welcome, Natalie. Thank you so much. This is the clicker thing.
Wow, not quite sure how to live up to this intro, but hi. It's actually, believe it or not, my first time at Nerd Nite, period, and I decided, presenting was where I should start. As Lauren amazingly introduced me, science fact or science fiction, we're going to talk a little bit about whether or not you should believe what you see in the movies. So just in case, high school biology was a long time ago, like it was for me. What's DNA? DNA is the blueprint of life. It is found in all living things. It contains all the information that you need to grow, develop, survive, and reproduce. And it is passed on from parents to offspring. So really, it's just really important stuff that you need to make you, you. And another little term, there you go. Come on, catch up. There we go. DNA sequencing is a term that I'm going to toss on. So DNA sequencing or sequencing is simply the act of reading the bases of DNA, so the letters, and which one are there and which order. So just reading letters. It sounds dumb, but it's actually really important for what DNA scientists do day in, day out. Really? Cops? Really? This is that bad? Oh, God. Okay. All right, so I hate to break it to you. When your mama said you're really special, you're really not that special. 99.9% of our DNA is shared with other humans. So everyone in this room, we have about 99.9% DNA that is similar. When we look at chimps, we have a whopping 98.8%. So even with chimps, we're pretty close. Cute little puppers. Does anyone want to guess what kind of percentage do we have? Our DNA is similar to a pupper's DNA by how much percent? I'm 70, 76, 85, just 95, whatever numbers. 94%, so we're pretty close. Whoever said 95, pretty close. What about a banana? 2%. 2%, no. 88, 45? 95? Oh, you can't be closer to a banana than a dog. Come on. Anyone else? All right, anywhere from 40 to 60%. So you're really like, when your mama says you're special, you're not that special. Except that you are. There are 3 billion bases or letters in the human DNA. So if we're 99.9% similar, that still means there's 3 million differences between you, at least between you and the person sitting next to you. That's what makes you unique. That's what makes you you. And it means you can do things like, there you go. You might be able to roll your tongue. For those who can't, I cannot do it. You might be able to drink milk without farting. You might be less susceptible to getting malaria, which if you live in Africa is a pretty cool thing, but you also might be more susceptible to have something like breast cancer. So, like I mentioned, we are going to be talking about movies and just how DNA is represented there. So, I'm going to go out on a limb and think that everybody in this room has seen Jurassic Park. If anyone hasn't and is brave enough to raise their hands, I will give you the plot of it. Oh, really? Sir? Really? You're doing this? Okay. Billionaire brings back dinosaurs. Dinosaurs eat things. And Jeff Goldblum is sexy AF. Like, that is, that's the plot of Jurassic Park. I really did not plan for someone to raise their hands, so I'm going to move on to this. Let's talk about the whole, like, dinosaur DNA is inside the mosquito, and then the mosquito is in the amber. So they get the dinosaur DNA out of it, and that process is called DNA extraction. Here's the thing. Getting DNA out of things tends to damage it a lot. Like, it is not a pretty process. It's actually quite messy. And so the idea that dinosaur DNA could live in a mosquito for a long time is kind of, So dinosaurs live anywhere from 250 to 65 million years ago. The oldest DNA that we have found is about 125 million years old. The oldest DNA that we have sequenced, which means that we get usable information out of it, is about 2 million years old. Most DNA, typically, if you reach really far, is anywhere from 0.5 to 1.5 million years old. So the idea that our dinosaur DNA lived long enough and is good enough to be able to create like dozens upon dozens of dinosaurs, I don't know about that one, guys. What I will say, and will point out, Luberay tar pits. If you don't know what it is, it's a little spot outside of LA, and it is a tar pit. So once upon a time, this big tar wave came in and trapped all these animals in it. Ancient DNA specialists believe that this is the biggest repository of ancient DNA in the world. Problem is, getting DNA out of tar is really, really difficult. So right now, scientists are saying, we can't get it out, so we're going to wait until there's a better technology. And when it does, oh man, they're going to have a field day about this. But yeah, so that is our DNA extraction. Next topic, CSI, crime scene investigation. For those of you who don't know, it is a show about crime scene investigations. It is. It ran from 2000 to 2015. Do not come at me. I know this is the OG. I know there's like 10 gajillion other versions, but this is what it is. The myth that I am not going to bust is that anyone would work in a lab this dark. No one in their minds works in a lab this dark. Just putting it out there. But what I want to talk about is DNA profiling, which is simply put, being able to match DNA from a crime scene to a suspect. So let's play Who is Smarter Than a Fifth Grader? On your left, you have your crime scene DNA. On your right, you have 3 different suspects. Can you find out which suspect whose DNA is on the crime scene? Shout out numbers. Did you say 2 and show 3 fingers? Okay, so what am I hearing? 2. Does anybody want to say anything else? No. Congratulations, you are at least as smart as a 5th grader. So you're like, yeah, guys, it's a match. Yay, we're all good. And I'm sure you're pretty confident saying like, dude, Natalie, seriously, like, I got this. What if I were to show you this much DNA? How confident now do you feel that it's suspect number 2? Or for my scientists in the room, if I show you this, how confident do you feel that it's actually suspect number 2? Probably a lot less so. You're not willing to bet your drinks on this, yeah. So it's definitely not willing to say it's a match. So, and then you have things like you have mixture samples. So you have the sample that you find on a crime scene. You know this is your victim, and you're trying to figure out which one is your suspect. And so there's this DNA. Forensic DNA is just a lot messier than what it shows on TV. And really what it is that 95% confidence intervals, which is usually what the standard for science is just not good enough for forensic science. Because people's lives are at stake, both the victims and suspects, and the whole, you're innocent until proven guilty. We work in 99% and above confidence. And so really, many of the samples that you find on a crime scene are deemed inconclusive. You're just not comfortable enough making a call on them. And it usually takes multiple samples to be able to get to where you need to be and get the answer that you need. So, Gattaca. Show of hands, who has seen this movie? It's both more and less than what I thought, so this is interesting. I'm going to go through the plot, and I'm going to butcher the plot. Please go see this movie. I'm not going to do it justice, but it's a really cool movie. Came out in 1997. So Gattaca is based on this kind of futuristic city and location where people are separated into 2 classes. The valids are people who've had their DNA enhanced so that they can kind of reach their full potential. And then the invalids are people who were born through good old mom and dad are getting together, after a few drinks. And so it starts with Ethan Hunt, our main guy. He's an invalid. His DNA is kind of like everyday Joe's DNA. But fortunately, he has tons of ambition. One of them is scoring this really hot girlfriend through Uma Thurman, who is a valid. Like her DNA has been modified and all this. And then there's Jude Law, whose DNA was modified so he can be a top athlete. But unfortunately, he has an accident and he ends up in a wheelchair. And really, Ethan Hunt, his character, makes a deal with Jude Law. And he takes his DNA. He takes his cells, he takes his hair, he takes his urine, he takes everything, and passes himself off as him. And then Ethan Hunt is able to have all these amazing opportunities that would not have been given to him. So clearly this is futuristic, but I do want to talk a little bit about what it would entail to have a society like this and whether or not it would have been feasible, especially and I'm going to put it out again, in 1997. So the premise of Gattaca, and like for a society like Gattaca to exist, you would have to have the ability to modify DNA. And that is kind of what we're seeing here. It's your pretty standard cut and paste or cut and copy and paste. So you take things out of the DNA that you don't want and you add things into the DNA that you do want. What does that look like in real life? So very quick history. DNA is discovered in the 1950s. In the 1960s, scientists start to mess around with it and figure out that they can do modifications or DNA editing. When you hit the mid-70s, scientists actually very smartly get together and realize that they're not sure that they can do modifications on human safely and reproducibly. They have these techniques, but they're just not comfortable doing that. And so they kind of say, we're going to pause on doing human experiments and modifications for now, and we're going to try and progress our techniques and be able to move forward. So things like synthetic insulin comes in the 80s, where that's a big push in the technology. Then in 96, again, very close to our Gattaca, Dolly the sheep, the cloned sheep that is 100% exact copy from its mother, I think. But that is actual cloning for the first time. And it's not until we get into like the 2010s that a new technology called CRISPR-Cas9, or CRISPR, comes into play. And this was the first time that we were really able to safely and repeatedly and smartly be able to do some DNA editing. So you can see that in 1996, something like CRISPR would not even have been on their mind. Like it's not something that would have shown up as something that was viable. So what can you do with DNA editing? You can cure lactose intolerance. So again, the whole drinking milk without farting. You can come up with a cure for cancer, which is really important. Fortunately, you can also go to the dark side. You can go into eugenics, which is driving populations and certain traits in populations. Or you can go into biological warfare. You can imagine having something as deadly as Ebola being as transmissible as the flu. That is a really big weapon. Or as some people thought for a long time, I don't know, kind of a long time, COVID was, engineered and something that came out of DNA editing. So this is what you would have needed to do the DNA modification. Then the second part of what you would have needed to do is being able to do DNA sequencing on a large scale. So in the movie, they actually take swabs, of doorknobs and hallways and everything every day, and they kind of check that the DNA of the people that are there it's what's supposed to be. And so to do that, just as a reference, so in 2001, one of the biggest projects to ever come when it comes to the DNA world was the Human Genome Project. It was unveiled in 2001, and what it was the very first time that human DNA was sequenced from the beginning to the end. Hugely revolutionary for anyone who works in the field. It cost over $1 billion at the time. It took over a decade to get done, and it took hundreds of scientists across multiple countries to get done. It was a huge project, a huge undertaking. But again, this would have been 2001. So by the time Gattaca came out, this would have been underway, still under wraps, like not super well known, but still maybe discussed. Move on to 2004. 2024, it's fine, guys, it's fine. Now, to sequence the human genome, so to look at human DNA, takes under 48 hours. You would be looking at 1 to 3 scientists. Does anyone want to guess what it cost? 30 what? Dollars. Was worth $30. Who else? $600. Okay, 3,000. Okay, we have some numbers out there. It cost under $1,000. So not quite 30. I wish we were there, but not quite. So clearly, in 20-something years, the cost of being able to do this came down drastically. I worked at the company that is responsible for bringing down that cost from $1 billion to $1,000 of what I do. With that, thank you so much for somewhat paying attention. I don't know if you have. I have time for a couple questions. I will say I will have to duck early, but please feel free to always flag me down if you see me at Nerd Nite. I love talking about DNA. Do you want to give a shout out to Kayla right there, who's the one who told me here? If you love the presentation, it's all me. If you hate it, blame her. Thanks, everyone. No questions? Okay. Ooh, okay, so out loud for the crew. Can you look at your DNA and find soulmates? Trying to figure out the polite answer for this. No, so genuinely, compatibility is something that you look at, like. Yeah, maybe not your soulmate. I don't know if, yeah. But maybe not your soulmate, but someone that you're compatible with. When you look at evolution, you wanted someone who's pretty different from you because we don't like inbreeding in general. It's just not something that we want to do. So you want to aim for someone who's kind of different from you. So yeah, you're looking at DNA. You're like, oh yeah, you're not too close to me. We probably make babies that aren't going to be weird, as opposed to your first cousin, whatever. But yeah, so soulmate, I don't know. But compatible, why not? Yes. What is my opinion of GMO foods? In the sense that humans have been doing GMOs for centuries because we want, the best stock of everything, and we get this really good cow with this very good bull, and we're going to get better babies. That, in a way, is you're driving GMO. You're picking up traits that you want. Afterwards, in terms of modification, all my answers are going to be like, this is why we can't have nice things. Scientifically, there's some really cool things. things that will, modifications that prevent droughts or make crops way more resistant to a lot of things. There's a lot of people who can benefit from it. There's also a lot of crappy things that can happen. So all depends on which side of the coin you fall on. The scientist in me loves it. The human in me, I'll leave it be. Oh man, dude, I am not nearly drunk enough to answer this question. There's a lot of work that's been, so the question was, the term is de-extinct animals, mammoths, dodos, any animals that are now currently extinct. There are people who are working on this. To quote very sexy Jeff Goldblum, just because we can doesn't mean that we should, or we were too busy worrying that we can doesn't mean that we should. There's a lot of work that's being done and a lot of people who are working on these things. Again, as the gentleman questions, the scientist in me loves it. The human in me, I don't know about that. All right, thank you so much, guys.
Thank you, Natalie. All right, we're going to take a quick break, refresh your drinks, stretch your legs, and we'll be back here in 10 to 15 minutes.
Hi, everyone. Welcome back. Thank you, first of all. I just need to say a second thank you because I looked outside again and it's still disgusting. And so I want you all to feel really good for leaving your house tonight because it's hard, y'all, when it's like this. And so you all just know that you get extra bonus rewards, good library karma, all of that. Thank you for coming. And already it's been worth it, but it's going to continue to get more and more worth it because we have 2 more amazing speakers tonight. I am so excited to welcome our next speaker, Paul. And Paul has had so many cool jobs, so you're going to have to excuse me as I look at my cheat sheet over here because I don't want to miss one. But Paul got his PhD from Columbia University studying the behavior ecology of monkeys in West Africa. So like maybe you were colleagues with Chris, the guy who started Nerd Nite, I'm pretty sure. Different universities, different animals, but in the same track. His snow leopard work began with the China Exploration and Research Society, and it continued as director of conservation with the Detroit Zoo, and continues as a regenerative farmer at the Detroit Flight Path Farm, and as a conservation technician with the Washtenaw County Conservation District. Like I said, all the coolest jobs. So I am so glad that he is here tonight. Please join me in welcoming Paul Buzzard.
Thank you very much. Can you hear me out there? Yay. Excellent. Okay. Advance this guy. Okay, tonight we're going to talk about snow leopards, this gorgeous beast down here. They're probably the most iconic species of the Asian highlands, Himalayas, but they're increasingly under threat from a number of factors. We're going to talk about 3 tonight. Climate change, construction, or better said, infrastructure development, roads, dams, that kind of thing, and COVID. But don't worry, leopards don't catch COVID. But because of COVID and the border changes and border shutdowns, it really affected the area that I work in with livestock movement. In fact, the area that I work in is really specific to the final 2, construction and COVID. But they're all factors for this guy. Overlap with leopards down there on the right, common leopards. Common leopards are about 150 pounds males versus 120 pounds for snow leopards. So they thought to outcompete them, but because of climate change, it's been shown around the world that it's getting warmer, forests are climbing up the mountains, so leopard habitat is increasing while snow leopard habitat is decreasing. It's been shown in many places. And these other 2 factors, the construction and the COVID, are sort of special for my spot in Nepal. So actually, let's go to Nepal. Here is Nepal. If you can see it down here, if you're not familiar, this little inset map down here, Nepal is tucked in between India in the south and China up here in the north. Just a really gorgeous place to work. I absolutely love Nepal. Incredibly diverse biologically and culturally because really within the space of 50 miles, 100 miles, you go from the hot, dry plains of India through the mid-hills, and boom, you're in the Himalayas. Incredible. It's like a 5,000 or more, or 10,000 rise in elevation. And the spot I work, again, here's Nepal, and Kathmandu, the capital, is right here, and I work right here. Here's Mount Everest on the border between China and Nepal, and I work right to the west of it, actually this little bit that rises up here, a place called Gauri Shankar, which we'll get to a little bit later. Now, just a bit more background about snow leopards. Like I said, they're the most iconic species. They're classic mountain species, and here's their range. This is again Asia. We've got Nepal right here. They're in most all mountain ranges in Asia. So the Himalayas is a stronghold. The Pamirs up to the Kunluns, up to the Tian Shan, all the way up to the Altai Mountains up in Russia. A bunch of snow leopards. But again, they are decreasing. Now for leopards, again, there we go. Common leopards. They've got a much, much bigger range. Incredibly diverse, adaptable species. Now this red, this is kind of where they used to be. So this is not quite this good anymore. But see, they used to range all the way from West Africa all the way up to Korea and Siberian forest up here. They go from grasslands, deserts, rainforest, forest, incredibly adaptable species. And here in Nepal, again, we're back in this area, a lot of leopards. And in various areas in Nepal, a couple areas in China, we're seeing, again, camera traps with both leopards and leopards and snow leopards. So we're thinking that's a really important factor in snow leopard conservation. Now, here's our study site. This is Gari Shankar Conservation Area. Again, here's Nepal, and again, this is an incredibly important area because it abuts, it's right next to two protected areas in Nepal. You've got the Sagarmatha, or the Mount Everest National Park here, and you got Longtong here. Here's Gari Shankar in the center, and also connects to a reserve in China, Chomolungma, which is the north face of Everest. Here's the area you work in, Lapshi area right here. And my colleague, my Nepali colleague, Narayan Koju, he was working there since about 2018, and he found the first picture of a leopard right up here in this Lapshi area, which is really he's really done the first systematic work there is done by my colleague. Years past, they found evidence of leopards here, but the first actual photo of a leopard was way up here by my colleague. So we got Gari Shankar area, which is incredibly important biologically and culturally for this reason. It's sacred. It's called a Bayul Valley. It's important for Tibetan Buddhism. And here's the main monastery, Gefoling. It's also a pilgrimage site for Tibetan Buddhists because of this gentleman here. This is Jetsun Milarepa. He is one of the most important Tibetan saints and poets. And some of his meditation caves are nearby here. He spent time there and got enlightenment. Potentially, Aaron Rodgers can go there also. So it's a really incredibly important area, culturally and biologically. So, really psyched to work there. And actually, back in the COVID year, I had some money from Nat Geo. Thanks, Nat Geo. Nat Geo Explorer Fund. So I approached my colleague. Here's Narayan down here with a blue sheep killed by a leopard, probably. But unfortunately, it was COVID time, so I couldn't travel, of course. But I had the money, so I sent it to Narayan, and I said, let's collaborate, use this money to find out just how important. I know you got the picture of leopards there, but how many leopards are there? How important is this valley for leopards? He said, great, let's do it. So with him and his students, he went there. He set some camera traps, and camera traps they've really revolutionized biology. They made our lives so easy. And here's him. I don't know how. Is everybody familiar with camera traps? Trail cameras? Okay, it's right here. It's just camera. You basically, it runs on batteries. You put an SD card in, take pictures, takes thousands of pictures, or tens of thousands of pictures, and/or videos. Basically, you set it and forget it. You plug it in, turn it on, strap it to a tree or a rock, and 24/7, night and day, takes pictures. When an animal walks by within 10, 15 feet, walks by, it senses their heat and motion, takes a picture. And you'll see some camera trap pictures soon. And also, they've come down in price a lot because hunters use them a lot. So they're getting very reasonably priced and making our lives as biologists really, really easy. So he went there in October 2020. He set 10 cameras. Him and his students walked transects. They found some leopard scats, which then they analyzed for diet. They saw a ton of blue sheep, Himalayan tahr, musk deer, which are really important snow leopard prey items. Also, they saw black bear. Just really a great study. And by now, here's a camera trap picture. It gives you the date, time, moon phase. Really remarkable tools. Okay, fast forward now to November 2022. And also, I should backtrack a bit. Back in this early work for my colleague Narayan, there was no reports of common leopards, just snow leopards in this Lapchi Valley. But by now, he's starting to hear word that leopards are coming into the valley, into the Lapchi Valley. So really interesting. So we went back there, and I was able to join this time in November 2022. We set 10 cameras. We collected some. And we collected some, also kind of a key with snow leopard urine as a lure to put some of the idea was put this urine in front of the cameras. Leopards come there, sniff it, stay there a long time. So we'll be able to see who is who basically, because leopards also make our life easier because they have spots which you can tell individuals who is who. If they stay there long enough and get good enough pictures, you can see if it's individual X, Y, or Z, and start to give you a pretty good idea of population size. So here again this is again Gary Shankar again. We're working, again, this northern bit that juts into China. Gives you an idea of where we set the traps and kind of the elevations we're at. We're down here like at 3,000 meters to 4,000 meters in the blue, up to 5,000 meters up here in the higher bits. And like I said, by now we're starting to hear that there are leopards moving in to the valley. So it's, exciting times. Oh, and also I was really keen to work. My colleague Narayan had been working with Nepali monks since he's been there. Nepali monks and herders. And I just was really keen to kind of meet them also myself and help work with them to help us with our camera traps, and in the future also with more scat collection. So yeah, really keen to meet the Nepalis. Okay. Oh, and also the snow leopard urine lure, as I said, this comes from my time at the Detroit Zoo. I love involving zoo animals or involving animals at the zoo in conservation projects in the wild, especially if you can involve actual leopards at the zoo. I mean, their urine is going to Nepal to attract other leopards. I mean, how cool is that? This is the Toledo Zoo. These are these three cubs here. 1, 2, 3. Almost see them. Their father gave me a bunch of urine. He was a good job. Also, Potter Park Zoo in Lansing. Their leopard also gave me plenty urine. Just kind of a sidebar. If now you have 20 some urine, 40 ml urine vials. How to get those into Nepal? Right. I mean, it's perfectly legal. I mean, I've got the email from Fish and Wildlife. It's an animal product, not an animal part. So it's legal. It's not illegal. But still, you arrive at Kathmandu Airport at 1 a.m. with vials of urine. You might have questions. So, anybody have any ideas how they would get urine in? Okay, here's the hint. The sock. Basically, I soaked socks. I soak socks in urine and then put those in bags and inside of bags and in trash bags, and no worries. and plus, you'll see from a camera trap picture, these socks were great to tie to trees or to tie, and plus I'm sure they held the scent for a long time. So if you ever want to camera trap leopards, urine soaked socks. That's your key. Okay, this is our first arrival in Kathmandu. Next morning, a couple hours of sleep. We have a day drive to the Lama Bargar in Gari Shankar. There's us leaving early. And actually, if you're keen to see this whole trip, go on YouTube and search Lapchi Valley trekking. Watch a whole hour-long video. Here's us in Lama Bargar. Actually, I mentioned the construction or the infrastructure development. This is what I'm talking about. This is a dam being built right here. One of 2 dams being built. So for a dam, you need the dam. But you also need a road to get the equipment there, and you got a lot of disturbance. So the idea was this disturbance is causing leopards, common leopards, to move up because they're being pushed out of their habitat. So that's one of the factors we're thinking might be happening. Okay, there's us on a really easy trail. There's me right there on a really easy trail, but it gets much trickier. Here's us. It gets a little sketchy here. Sketchy here too for a different reason, but yeah, a little sketchy there. Little sketchy there too. It does get bunched, but now we're getting close to the monastery and Lapchi, the summer houses. These are kind of the winter houses of the Tibetan herders. See all the yak down here. At fall winter house of a Tibetan herder. And here's where we stayed. We stayed down here at that big monastery. Now we're just, we're right next to one of those meditation caves of Milarepa, and we're just having a nice look down upper Lapchi Valley. Okay, so what did we find? We found a ton of leopard prey. Blue sheep, which is basically their favorite. These wild goats, Himalayan tahr, and musk deer. I don't know if you know musk deer. Musk deer are awesome. They don't have. I mean, they're not true deer. They don't have antlers. They've got tusks. You see those tusks? Oh, they're so cool. And this place is really special because musk deer are really heavily hunted. I mean, the musk pods in the males it's worth its weight in gold. So they're really, really heavily hunted. But here, this was taken with a camera, with a phone. They can see them right from the trail. So that's our next work map with musk deer, because they're so cool. Okay, and also this is what you came for, though, leopards. We got snow leopard. We got there's one sniffing a sock right there. See, it works. Thank you, Potter Park, and thank you, Toledo Zoos. So we got at least, and again, we got good pictures. We got at least 6 adults, and actually this one here, he's got like his eye is all manky and messed up, so he was really easy to tell him at night and day who he was. Otherwise, at least 6 adults, I think 2 or 3 young or juveniles. So really, a ton of leopards. We were thrilled, and 2 common leopards, or regular leopards down here. So, yeah, we're starting to see, the overlap and the potential competition, which we were worried about. And what does this mean? Don't worry so much about these graphs, the details. These all 4 graphs have the same X and Y, and they're all basically showing activity patterns of leopards and snow leopards. With on the X-axis, you've got time of day, just from midnight to midnight. And this Y-axis is kind of a measure of activity pattern. Basically, when did the cameras take pictures? Was it in the morning? Was it in the daytime? At night? That's what this measure of Y is. So let's start down at the bottom. You've got snow leopards over here on the left, and leopards. This is when they're alone. The camera's only taking pictures of leopards or snow leopards. You see, they're both about the same, or similar. Both active in the morning and in the evening and at night. Morning and night. But we get up here to top left. This is 8 cameras that had pictures of both leopards and snow leopards. And this is leopard. He's here. But snow leopards, they seem to have moved. And this is where they overlap. Snow leopards, though, seem to have shifted their behavior later in the morning. And they're still both overlapping a lot in the evening. Now, this is probably the most important one. This is showing snow leopard behavior before leopards. Remember I said, like, my colleague Narayan, they had no reports of leopards, and we didn't started getting pictures of leopards until like 2023. So we can look at snow leopard behavior before and after leopards arrived. I mean, I'm not sure if they quote unquote arrived, but it looks like it. So you can see before leopards, before leopards, they're up here. Then after, they're way down here, and they shifted all the way to the evening. Now, is this because the snow leopards are being displaced by leopards? We don't know, maybe. And this whole thing is truly a work in progress. So I'll have to give you an update in a couple years. But we have pretty good evidence that they shifted their behavior at least. And now just to get back to my sort of big question, which of these is the most important? Well, climate change, that is the most important. I mean, that's everywhere. The forest is rising, and the habitat of snow leopards is decreasing. As for the infrastructure development, the dams, that's a good question. Well, we'll see. I mean, now the dams are finishing up, or will it finish up in a couple years, and we'll see if once the construction stops, maybe there's less disturbance. Maybe if leopards come back down in elevation, we'll know. And COVID, let me just talk about this a bit. Now here's Agari Shankar. Again, it's surrounded by, this is Nepal, but it's surrounded by China, all around it. And in fact, until the 60s, this was part of China. But, I mean, for some reason, well, because of the war, China and Nepal fought a skirmish, a little war. And for some reason, China won, but yet they still gave this Gari Shankar Valley to Nepal for some reason. And so the herders here, these Tibetan herders, became overnight Nepali citizens. But they've got special arrangements so they can still drive their yak across the border like they have for thousands of years. They can still drive their yak across the border to trade with Tibetans in Tibet. Except COVID, right? Border shuts down for 2 or 3 years. The yak keep reproducing, and so the yak herd built up. And I think that might be one of the things that really brought the leopards up in elevation to feast on these yak and horses. But we'll see. And now I've heard, now that my colleague Narayan, he says that now the border was open, they sold almost all their yak. So it's really a place in kind of in flux. So it's interesting. And finally, Like I said, it is very much a work in progress. So I'll be going back hopefully in July, thanks to funding from Panthera NGO. Thank you. Anybody from Panthera here? Didn't think so. But yeah, I'll be doing something like this actually. I'll be doing community meetings with the herders and the monks. We'll be showing them pictures of what we found and what we plan to do. More cameras. Thank you, Panthera. We're gonna have like 40, like a lot more cameras in a lot, up, lower elevations, higher elevations, much bigger spread, and importantly, we're gonna do a lot more scat collection or fecal samples. We're gonna get a bunch of fecal samples, so we'll be able to do a data analysis, and with DNA, we'll be able to tell. Yay! Thank you. Did that scat come from a leopard or a snow leopard? Awesome. And look at the bones. What are they eating? Are they overlap? That's gonna be the big paper right there. What are they eating? And plus DNA. Thank you. We'll be able to tell did the scat come from individual A, B, or C? So we'll get really, really robust information on how many leopards are there, how many snow leopards are there. So it's exciting. And with that, special thanks again to Toledo and Potter Park Zoo, and thank you very much. Two questions? Okay. 2 questions. Yes. Right. I wish we knew. I mean, and actually, that's I forgot to mention. But on some cameras, we had a snow leopard. I forget who was first. We had a snow leopard. Then half hour later, a common leopard. I mean, so it was within 30 minutes. Incredible. And must have still smelled his or her tracks. I mean, the idea is that the snow leopards are scared of leopards because they're that much bigger. They're 30, 40 pounds bigger. But I kind of want to think that if also I should have mentioned with the herders, when we go back in July, we're going to talk to them. They got rid of most of their livestock, and there's a road built there now. So we're going to ask them, hey, how about ecotourism? And maybe find a way to put a value on the leopards, and to keep their livestock herds down, so that the blue sheep and everybody else can be abundant. So there's enough food for both common and snow leopards, and the herders are happy, and we'll work with them to have sustainable, low impact ecotourism. That's sort of the dream, and hopefully people will be able to see, musk deer for sure, and Himalayan langurs, and blue sheep, and potentially snow leopards. So if not, they could at least see the camera trap pictures, which the herders will have. Sorry, did that answer your question? But I think the snow leopards, the idea is they'll be. We think they're being pushed out. The data is just on the timing of the cameras. That's not the best data in the world, but that's what we had to work with. So the diet overlap will tell us a lot more. Time for one more question? Yes. Well, potentially. They're not like ligers. They could, but they're actually fairly. What was that? Tigers more closely to snow leopards, and leopards are more closely related to lions. So they are fairly far apart. They're both Panthera, both the same genus, but they are fairly far apart. So I like to think so, but I don't. Well, with the cameras, with the videos especially, if that comes up, that'll be my science paper. Thank you.
And thank you all. We're going to take one more 10-minute break, but definitely come back because we've got to figure out a way that this third talk will also relate to soulmates. So think on it. See you in 10. The music has faded because it is time for your final Nerd Nite talk of the night. I know, it's exciting stuff because we are continuing our killer lineup tonight with Katie Furman. Katie is a PhD student in neuroscience at the University of Michigan, where she uses optogenetics and mouse behavior techniques to study the neural circuits involved in food consumption decisions, which is really cool, or at least I think so. But in her free time, you can find her picking up an impossible amount of sci-fi and fantasy books from the library, which I love to hear. If y'all don't have your library cards yet, but live or work in Ann Arbor, you should get one, because don't pay for your books, get them from us instead. And going on long walks with her Chihuahua, Edison. And if you know of any job opportunities in science communication, there's someone who might be in the market for it, and you're about to see her demonstrate her skills. So it's a pretty great night. Join me in welcoming Katie Furman.
Hey, can people hear me okay? Awesome. All right, hi everybody. I'm Katie, and my talk is called Mouse Mind Control, how neuroscientists use lasers to activate brain cells and solve science mysteries. I'm a PhD student at the university, and before I get too deep into it, I wanted to kind of just give you guys some of my credentials before I get started. So about me, I grew up in central New Jersey. I have a tiny chihuahua named Edison, which was mentioned in my bio. He's pictured over there. I love reality TV. That probably has to do with my New Jersey roots. Similar to the snow leopards, I also hate climate change, and I am a neuroscientist, which is probably of the most relevance to this talk. In my day-to-day as a neuroscientist, my research focuses on how our brain is able to understand the food around us. So not necessarily hunger, but something along the lines of like how you understand the value of this food versus that food, and how you make the decisions regarding what specifically you want to eat. And in order to study that, I use a technique called optogenetics. And I think it's the coolest thing ever. I think it's like the coolest science technique that's ever been developed, and I want to tell you guys about it. So just like before I can get into the nitty-gritty of how this works, there is some context to set up so that you can fully appreciate the beauty of this technique. So you can pretty much summarize all of biomedical research with just a few words, and that is fuck around and find out. That's what we're doing on a day-to-day basis, myself and my colleagues in neuroscience and other fields. We're fucking around and we're finding out in different and interesting ways depending on your field and your specific topic. But this is something that, as you may know, all of us are doing on an almost day-to-day basis, is fucking around, at least, sometimes finding out. And I just wanted to show you an example of what I mean by this. So I have a video, courtesy of a Reddit user, of some kids doing exactly what I'm talking about, fucking around, finding out, and learning something new. So I'll just play the video. You can tell that this is the first time any of these kids have ever walked out of a tent. Because they don't know how to do it. Every last one of them. So we're learning, right? They're learning here. They haven't done this before, and they just learned something valuable about their environment. But you may have noticed, you may have missed this the first time, so I want to replay the video. I'm sorry if the slides don't like it when I try to do that. I think it's going to work. Okay, we're good. Focus on the last kid, because all of the first four kids trip and fall in earnest. The last kid's just going along with it. So he trips, watch him. He carefully steps over and then falls. And that's because he's learning, right? You can't hear the audio to this video, but what's going on is it's funny. And the parents are laughing and all the people around them are laughing. And so that last kid's watching, observing his colleagues fucking around, and makes an informed decision of like, I don't want to trip and hurt myself, so I'm not going to. But I know that when I fall, it's going to be funny and everyone's going to enjoy the area of their brain is damaged. And then a clinician is going to find out based on that damage and the deficits that result, like kind of working backwards to figure out, oh, that part of the brain must have been involved in speech because now my patient can't speak. And so that's physical lesions. I mean, it is definitely a fuck around, and it's definitely a find out because, a whole chunk of the brain is gone. But there are some limits to this technique, which is that you really can't go back. Once this is done, whether it's done intentionally or not, you can't fix it. You can't put the brain cells back. And so you're limited in what you can study this way. Additionally, if you're taking a brain area out intentionally, you really can't be sure exactly what you took. I might be interested in, say, like one really specific region of the brain, but I might not be able to isolate exactly that part. I might have to take with me like some of the surrounding parts and other connected areas. And so you can't be sure exactly what you're looking at when you're studying the brain this way. So another way of studying the brain is to electrically stimulate neurons. And what I mean by this is literally injecting electric current into a brain cell. And that's the way that our neurons are communicating anyway, is by using electrical signals to send and receive messages. And so this mimics the way that your neurons are communicating anyway, but it is not without kind of issues in itself. One of them is that this can be kind of dangerous. If you're not careful, you can have your parameters wrong and really damage the cells. And you're working with like electricity, I don't know, you could damage who knows what. I don't do this. But, and then you also can't be specific to a certain cell type. And so you may have heard of like dopamine and serotonin. These are specific neurotransmitters that your brain cells are going to be releasing. And a given brain cell is, for the most part, only like using one of those neurotransmitters. So maybe I'm really interested in dopamine, but not serotonin. I can't pick just the dopamine cells when I'm using this technique. I have to pick just like an area of cells. And so moving forward, another technique is pharmacological manipulation. And drugs. That's what that is. So injecting a drug into a cell. Examples, Zoloft, Ozempic, and Adderall I have on the screen. You may have heard of those all in the news recently. This might be a little bit more self-explanatory. Giving a drug to a subject, whether that's a lab animal or a patient, and seeing what the outcome is, right? And this is really important work. This all is really important work. Like, we would have no idea how to use any of these types of drugs on like suffering human patients without the animal work. But again, this comes with limitations. One is that once the drug is in, you can't take it out at will. So, you know, if you take a medication, or if a lab animal is given a drug, they've taken it and now it's not going to be out of their system until it's naturally metabolized and they pee it out. And so once the effect starts, you can't stop it on your own. Additionally, you might not know how long it lasts. And so if I need to look every 2 hours, I might not know if the drug is already out of the system by 2 hours. And so I might need to take way more time points, or I might need to do a lot of tests to figure out how long the drug is going to be around. And lastly, you can't be sure if you didn't cause side effects. And so I could administer a drug hoping to look at, for example, like increased focus with the example of Adderall, but I might also cause, cardiac and respiratory effects that are gonna make my patient like way less focused because their heart is beating super fast. You can't be sure that these side effects aren't happening when you administer this drug, and so it's somewhat messy to study in this way. So, I've talked about physically ablating neurons, electrically stimulating neurons, and pharmacological manipulation. These are all super useful techniques and are still used like every day today, but they all have their own faults, which is why I want to get into my personal cup of tea, optogenetics. And so optogenetics comes from the words opto for light and then genetics for gene expression. And so we're using these two things, light and gene expression, to manipulate the activity of neurons. And the great thing about this is you can turn light on and off with the light switch. You can literally flip this on and then flip it off. It's as simple as that. And so when I was talking about the timing aspect of it, like if I'm using optogenetics, I can literally just turn my effect on and then turn it off and see it like that, light speed, if you will. However, I just told you I'm gonna use light to activate brain cells. The brain is famously inside the head. Typically, light is not getting in there under normal circumstances unless something's gone really wrong. And so what this means is that, one, the light's not getting into your brain. The light is not touching your brain cells to begin with. But even if it did, your brain cells haven't developed any way to be able to read that, to like see that light's coming in, because they've literally never seen the light of day. Your skull and your skin and your muscle has always been covering your neurons, and so they have no capacity to sense light whatsoever. So I brought up two problems. If I'm going to convince you that optogenetics is going to work, these are some problems I have to solve first, which are that light doesn't normally reach brain cells, and even if it did, those brain cells don't have a way to respond to it. However, conveniently enough, we do naturally have a cell type in our body, or an organ in our body, that does respond to light, and that's the eye. You may or may not know this, but that's what vision is light. You're seeing different light waves and photons hitting your eyeball, and from that, your brain is able to perceive all sorts of visual stimuli from the world around you. So you are already naturally able to perceive light, just not in your brain cells. So I'm going to go through how this works in the eye, and then how we could use this to work in the brain. If you zoom in on the retina of your eye, there are these proteins that are called rhodopsins. And these rhodopsins are like at the cellular level. These are the things that allow this response to light. And so what that looks like is the light comes in, the rhodopsins are activated, and then they send their signal into the eyeball, then into the neurons and into the brain, and convert that literally light penetrating your eye into an electrical signal that your brain can understand. So this is what's going on in the eye. That's not what's going on in the brain. What's going on in the brain is you have neurons. You may or may not be familiar with neurons. They kind of look like this. There's going to be like a message will kind of originate here, and then it'll send it down there onto the next neuron, and it's like wires that are connected to each other, and that's how they communicate. And so I'm showing you this pink neuron that might receive input from a blue neuron and might give input into a green neuron. And how this is going to work under normal conditions is that if by some crazy like turn of events light does reach your brain cells, nothing's going to happen. That pink neuron doesn't have a way to be able to respond to the light. But if I take these same rhodopsins that are in your eye and I can genetically target them to your neurons, then all of a sudden, let there be light. There's a way for this specific cell to be able to hear the light or sense the light, do its thing, send that signal onto the green neuron. And you'll notice here that the rhodopsins are only on this pink neuron. For the sake of the presentation, pink neuron is a type of neuron. But earlier I used the dopamine example, or serotonin. With this technique, you're able to genetically target the identity of the cell. So if this is a dopamine cell, I can specifically express those rhodopsins in the dopamine cell and not the other cells. In this case, the pink cell and not the blue or the green cell. So one problem solved. Earlier I said that light doesn't normally reach brain cells, and I said that if it did, those brain cells don't have a way to respond to it. Well, I just gave them a way to respond to it, so that's settled. Now we have one other problem to tackle, which is that light doesn't normally reach brain cells. So how we're going to change that is a little bit easier. Imagine here's your little mouse. If we're working in the lab, this is being done in mice or rats. That is what its brain looks like from that kind of from this view. And if I'm interested in that specific area of its brain, imagine I take the cross section. Now I'm looking at it like head on, and I'm interested in that specific area. I don't know if you can see the pink that just lit up. And so if those pink cells that I was talking about on the last slide, if they're located in this specific area of the brain, what I'm going to do is implant an optic fiber into specifically that area of the brain. And so this is kind of like almost like a flashlight, like it's a really long and really teeny tiny light bulb that we're able to connect to a light switch and to a laser that has the light in it, and you can literally turn it on. So the mouse looks like this with its optic fiber implanted into an area of interest, and you can see this blue light because blue is typically the color of light that we use for optogenetics. I can get into that if people have questions about it, but this is what your mouse looks like then when you've implanted the optic fiber, and you're now able to deliver light into its head. And so now both problems are solved. Light doesn't normally reach brain cells, but I have just explained in a way that it will. So to recap, before I get into the really exciting part, we've now got a mouse that has a way to deliver light into a really specific target area of the brain. And that that really specific target area of the brain has these rhodopsins in a really specific target cell type to where we are able to specifically turn on a light switch, activate that pink cell. Now, putting it all together. In 2013, this research group, the lead of which was Dr. Josh Jennings, I don't know him, but he's the lead on the paper, they had a reason to believe that VGAT neurons in the BNST play a role in hunger. So VGAT is the type of neurons. In my last slide, it was a pink neuron. This time it's a VGAT neuron. It's just like the identity of the type of cell that it is. BNST is the brain area. It stands for bed nucleus of the stria terminalis, but everyone just calls it BNST, so we'll call it BNST. So they have a specific identity of cell in a specific area that they think, for whatever reason, is going to play a role in hunger. So what do? They take their mouse, they find out where the BNST is. Turns out it's like roughly there. My little pink didn't show up as well on this slide. They implant their optic fiber, and they have the rhodopsins in those VGAT cells specifically. So again, the mouse looks like this. And now I'm going to show you a video of how this all works together. So before I play it, I kind of want to orient you to what we're looking at here. So this is a bunch of mouse food. This is a mouse. This food specifically is like this high-fat food. The mice really love this stuff. They like it so much better than the normal food. So they're pretty motivated to eat it, is what I'm saying. And then here's your mouse with its head cap that I showed earlier, just kind of a little helmet to hold everything in place, and the optic fiber with the blue light on. And so what's going to happen in this video is he's just hanging out. They turn the light on, and this mouse immediately starts eating. The instant the light turns on. Can you see that the light turned on? Yeah. So he's eating, munching, chomping, loving it. The light turns off. He literally drops it, goes to do something else. That's like instantaneous when the light turned off. And off screen, there's literally an experimenter with a switch that they're flipping. The light turns back on. He picks up that tiny crumb, eats it, goes back to the giant crumb that he had before, eating it, chomping it the whole time that the light is on. This time's longer. I don't have enough words to fill the time that he's going to be eating. But yeah, just wait till it turns off. It is instantaneous. Still eating. It's off. Doing something else. And again, that's like this high-fat food that they love. They should be like motivated to eat it. And so the fact that he starts eating it the second the light turns on is crazy, but the fact that he stops is also crazy because it's really good stuff. Imagine like a toddler with candy. Like to stop eating it when the light turns off is just as fascinating as to start eating it when the light turns on. So if you've been paying attention, following along, you may think I've just explained to you how we can experimentally do mind control. I've just shown you that with literally the flip of a light switch, we can turn brain cells on and off in the laboratory. There are completely valid reasons to be wary of this, and I want to address that before I get off the stage here, and explain why we don't need to be scared, why this is just cool. First of all, mind control, as you might think of it in the case of like science movies and stories and stuff, is definitely not possible with this technology. So like the idea of coming up with the thought even is something that neuroscientists don't really understand yet and involves so many different cells and different like neural circuits. And with optogenetics, we can really only target one or two at a time. And so it's really not going to be possible to ever like generate a thought or a consciousness like within an animal this way. It's too specific, and we just don't know enough about how thought and consciousness work overall, let alone have the tools to do that. This is also an experimental technique, but not necessarily a therapeutic one. And so you might be thinking like, you know, turning on brain cells that don't work, like maybe that's a really useful tool. But and it could be, but not really in the case of opto, because it's like taking a giant sledgehammer to a case where you only need like a tap of a finger. We're turning on these cells at a super high degree, like way more than they ever are naturally being turned on. And so while that's really useful in an experimental setting to understand how stuff works, it's not going to ultimately be that useful if implanted into a human person to try and solve a disease. And so it's pretty much irrelevant to a lot of disease treatments. And this will not get FDA approved without a therapeutic reason for it. And I've just explained that. I don't think there's a therapeutic reason for it, and so we will not be seeing this happening in humans anytime soon, not in any of our lifetimes. And so I hope I have expressed to you why I'm so excited about optogenetics, why I think it's the coolest technique ever. And with that, I have a couple of acknowledgements. First of all, this is Ed Boyden and Carl Disseroth. These are two of the guys who are widely credited with inventing optogenetics, so big ups to them for giving me a topic today to discuss. I want to also acknowledge my dog. He was sitting on my lap while I made these slides, keeping me warm, and he's just a super cool guy. If you guys ever meet him, you should say hey. I want to also acknowledge all of the research animals that go into developing a technique like this. This is a statue that's in Russia. It's called Monument to the Laboratory Mouse. It has a little plaque underneath it that just like acknowledges the sacrifice that these rodents take so that we're able to make these huge scientific advances. And that's like really, really important to the work that I do. And so acknowledgement to their sacrifice as well. I want to acknowledge my lab. These are some of the people that I work with on a day-to-day basis that make this work really exciting and collaborative. I want to acknowledge the Michigan Neuroscience Institute where I work. And lastly, and most importantly, I want to acknowledge Nerd Nite and the Ann Arbor District Library and Live for this incredible collaboration that y'all put on. I love Nerd Nite. It's such a fun thing that we all have access to. So with that, thank you, and I can take questions, I think. I can take one question. Yeah. Yeah, so I showed somebody else. The question was, how do I use it in my research? I showed somebody else's video, but what am I doing? I use somebody else's video because it's way cleaner than what I do. It's pretty obvious, the effect that you turn it on and they immediately starts eating. I'm using it in a little different way where I said I was studying like the valuation of food. I study a subpopulation of neurons that is involved with kind of the understanding of food value. So like if I turn on these neurons, it doesn't make you hungry, it doesn't make them eat like that, but it does over time seem to create an association with the food that they're eating to make that food better. So essentially what I'm doing is like giving the mice the choice between two different foods. Spoiler alert, it's the same food, but one of the foods comes with this optostimulation, and I'm observing over time whether they like choose to prefer the stimulated food more than the other food. And this depends on the brain area and the type of cell, and so I'm doing a couple of different ways of like combining all that to understand the way that the way that mice value food. Can I?
Live has another show tonight, guys.
Sorry.
So answer quick, and then everyone go settle your tabs.
Yeah, you can follow me on Twitter too, or DM me on there. I put my @ if anyone has more questions. Thank you.
One more time, Katie Furman. That was excellent. Thank you all so much. Thank you to Paul Buzzard. Thank you to Natalie Catchings, our other speakers tonight. Thank you. I wanted to say two quick things. No Nerd Nite next month. I'm sorry. So we're taking a little hiatus. We always do this in May. We always do this in December. But you can catch us at Top of the Park in June. So mark your calendars for that, please. And then we'll be back here in July. All right. Good night, everybody. Thank you.